Welcome to Basic Hematology. Today's video lecture will be consisting of two separate presentations. The first presentation, it's just a very brief introduction to hematology. I'm going to introduce you to some few key words, lay out how the course is going to be ran, and some tips and tricks that might help you along the way. The next lecture, we are going over basic concepts in biology. So if you are not comfortable in biology, then this should be a pretty good way of catching you up. There's some key ideas. I'm not necessarily saying that you need to memorize everything in that, but there are key ideas that I would like you to know moving into the next few lectures. So let's just do a brief introduction to hematology. So here I have some objectives. At each lecture, I try to have objectives that I hope will help you in exams going forward. Just sort of keep these in mind. If you are able to achieve these objectives, then you will be on a very good path on being able to pass your exams and understanding the key terminology going forward. So, hematology. What is hematology? Well, heme means blood, tology is study. So, therefore, hematology is the study of blood or blood forming organs. We also call them hematopoietic organs. Hematopoiesis is the term for blood formation. Blood has always been a primary fluid ever since the Greeks thought of the four humors back in 400 BC. It's a very old, outdated way, and it's sort of kind of true, where the Greeks observed that there would be four fluids coming out of the body at a given time. So they had the yellow and the black bile, phlegm, which is your spit, and blood. Though now we don't go off of the four humors. I don't think we have any modern day plague doctors, but we do realize that a part of it is true, is that the Greeks believed that the balancing of the four humors was key to living a healthy life. And that is sort of true if you think about it in that way, that any sort of altercations to the components of blood can be a result of disease, but not necessarily the primary cause of it. So then, sort of going forward from the four humors, we got better and better at understanding our body, and now we know that hematology is vital to help determine any active infections or needs to transfuse a patient with any various blood components like platelets, red cells, obviously. um, but we wouldn't be able to do surgery without having hematology, and we wouldn't be able to have blood bank if we didn't understand that we needed red cells in the first place. So this is the basic course breakdown. Obviously, if you're not attending my course, then this isn't going to help you. But for the students who are attending my courses, the first exam, we're just going to go over basics. Where I just need you to understand the terminology, and I need you to understand how cells develop and grow in the first place. The second exam, we're going to start getting into more technical and specific terms. So we're going to look at the blood cell development and the stages. Third exam, this is where you learn how to work in a hematology lab. This consists of lab testings, their meanings, and their troubleshooting. And last but not least is an introduction to anemias. It is just one lecture. And hopefully this will help you set up for advanced team where you learn the specific diseases and how to identify those diseases. But the two biggest tips that I have regarding my course is this. First is try to understand and learn key concepts. You are going to put yourself through agony if you try to memorize everything. I promise you, you won't have a very good time if you don't understand what things mean in the first place. If you understand what terminology means and what the terms represent for one thing or another, the better. If you can develop on that understanding and be able to draw conclusions based on hypotheticals or problems, then you will do excellent. And also, remember, I am going to be using the terminology that we use all throughout this course. The basics will not go away when we're done with that lecture. So be sure to try to make a habit of reviewing previous lectures occasionally. And if you can't get from the basics, then they only continue piling up. So in order to get from the bottom of a pyramid, you have to build those lower levels really strong in order to climb your way towards the top. So that will be really helpful, especially in advanced team. So the composition of whole blood, it's very cool. So this is what your blood will look like if we just filled it out in a tube. Well, in order to get separate each of the components out, then we have to centrifuge it. Once that tube undergoes centrifugation, your blood will look like this. So you'll have this liquid portion, which is called plasma. That is the liquid portion from unclotted whole blood. And at the bottom, where it's still red, will be your formed elements. Your plasma is made up majority of water. If you haven't known, your whole human body is made up of majority water. Same thing as with your plasma. Then we have some proteins floating around in there, and we have some other solutes as well that are also vital to your well-being. And the formed elements, and this is where hematology really thrives, we have three key components. It's your red cells, which make up millions. It is by far the most numerous cell in your blood, followed by your platelets, and last but not least is your white blood cells. Your white blood cells are made up of cells called neutrophils, lymphocytes, monocytes, eosinophils, or basophils. And don't worry if you have no idea what any of that means, we will be going through it through this course. But just understand that your whole blood is made up of two components: plasma, which should be about 55% of your whole blood, and your formed element, which is 45%. Um, Now, that being said, I do just want to note that plasma does make up approximately a 55 of your blood volume, though this could fluctuate a little bit, especially within populations and even across the sexes. Um, but know that when I say like approximately, these are good numbers for you to memorize. These might be plus or minus 10 at the most, again, depending on your demographic. But just most people, plasma is going to be 55%, formed elements will be 45%. Um, this is what I want you to start getting used to, being able to swap back between these terminologies. So when I say erythrocyte, I'm talking about your red blood cells. When I'm saying leukocytes, I'm talking about your white blood cells. And if I'm talking about thrombocytes, I'm talking about your platelets. I may use these interchangeably throughout the course, but I just need you to know the formal terms for all three of these elements. Now, not only does this composition between your plasma and your formed cellular elements depend on your demographic, very notable variations to these are also good signs of any signs of disease as well. If I start losing blood, then I'm not going to have 45% of my cellular elements anymore. It's probably going to drop down into like, if it's really bad, 10%, but let's say 30%. Um, so, or if I'm having a huge amount of formed cellular elements, then I'll have less plasma, which the both are also a problem as well. So even just knowing that, that's good for you to know. An average adult also has 4 to 6 liters of blood. Most people, you shouldn't be losing more than that. Also, I just have this in italices here. In hematology, we don't really talk about this too much, but in clinical chemistry, we talk about this a whole lot more. So, serum is also your liquid portion of your whole blood, but the serum does not contain any clotting factors, and serum is also formed as a product of whole blood clotting. That's why when you hear people talking about giving something in blood bank, they'll always say plasma. They'll never say your serum, because if you gave somebody your serum when they're low on platelets, it's going to be useless to them. Again, the primary component of plasma is water, and again, within the plasma, there are these dissolved elements. All these are crucial for you to live. Um, of course, this is hematology. I'm not going to expect you to memorize all these off your bat, but if you are in my clinical chemistry course, you will have to eventually learn these. The main function of blood plasma, though, is to act as a transport system. If you ever threw something down a stream of water, you know that water is very good at carrying things, and that's what plasma does. By acting as a transport system, it can help out with cell metabolism as your body exchanges nutrients and wastes. Right, for your blood cells or your formed cellular elements, every single one of these have to be formed from the bone marrow. Of course, when they're little itty babies, they are undifferentiated, which means they don't have a function yet. So they're going to continue to multiply or proliferate, and then over time, they're going to develop or differentiate using different cytokines or signals to help become different blood cells. Once they are mature enough, they go from the bone marrow and into the peripheral blood. All right, so there's three cellular elements like I talked about before. These are their names and these are their functions. Erythrocytes are your red cells. Their primary job is that they contain hemoglobin. This hemoglobin then transports oxygen and CO2 and acts as a exchange between the two in your lungs and in your tissue. Leukocytes help protect your body from anything that's considered non-self. Big three is bacteria, viruses, cancer, and if you're living in other countries, parasites. And again, these are made up of your neutrophils, eosinophil, basophils, lymphocytes, and monocytes. Last but not least are your thrombocytes. These are tiny little cell fragments that come from a bigger cell called a megakaryocyte. These fragments are vital in you being able to have clots. Imagine if we did not have clots, you and I would be bleeding to death from microabrasions all the time, and it would suck. By the way, the formal term for blood clotting is called hemostasis. This is sort of a really important point when you start working or if you're interested in becoming a doctor. Um, we have reference intervals, or basically the things that tell doctors when they're looking at the results how much they fall within the population average. But none of these three cellular elements of blood, or even the composition between plasma and red cells, are not equally present in everybody's blood. They can vary by race, age, sex, geographic location, diseases, or even your time of day. Whoa! So, because of that, individual labs must come up with their own reference intervals for that lab. This helps gives a point or determines normalcy within a geographical area. So usually this is based on a confidence interval of 95%. So and most people will fall within this sort of average within plus or minus 2 SD. I just need to stress that not every single reference interval you're going to see in my class is going to be universal. But the ones I am going to give you are the ones that they're going to quiz you on your boards. Sorry. But just understand that people are diverse, and because of that, we need to come up with our own ways to be able to help tell doctors for the population that they are treating whether or not they fall within normalcy. For example, and I'm just only bringing this up because I work at a pediatrics hospital, neonates will have a much higher red cell count and a white cell count than any other age, and that's normal. They're babies, they're growing. But once they sort of develop after 6 months, then they'll start falling back within adult parameters um, until you're about 4 or 5. Majority for adults, by the way, the majority white cell in your blood is going to be neutrophils when you're at an adult's age, but when you're a kid, most of them are going to be lymphocytes. This is also known as inverted differential. course, once you turn 5, then you start looking like an adult, closer-ish at least. Um, I'm not expecting you to memorize all this now. I will continually reference back to this, but this is a pretty neat example of just how your age alone can help determine what is normal for you. All right. Like I said, hemostasis is the formal term for blood clotting or the ability of your blood to clot to help prevent blood loss. Um, that way, your body will clot at the right time. Your blood will remain as a fluid when we want it to remain as a fluid, and when we need it to form a clot to help prevent blood loss, it'll do that. Um, so we have a lot of screening tests. You'll hear the terms PT and PTT. These help test our primary hemostasis. Um, or sorry, so the initial clot in any sort of vasoconstriction assay, or how well does your walls constrict, that's called primary hemostasis. The formation of fibrin that helps stabilize the clot through the coag cascade, and any other factor assays help evaluate your secondary. I'm not. This is not coagulation class. I'm not going to go through this, but you will have to know it for your boards if you're planning on taking it. Just understand that the proteins that are involved in this, if any one of them are affected, then that can result in disease states. If you're clotting too much, then you're going to risk stroke or having an emboli or just a big clot that ends up in your heart. Or if you're not clotting enough, then you risk bleeding out and it sucks. Since you are presumably going to be a laboratorian, if not, then this will be a fun fact for you. Um, Lab testing is going to be divided into three components. First is your pre-analytical, second is your analytical, and last is your post-analytical. Pre-analytical tests all testing aspects before we get the ability to be able to run in the first place. This is including your blood draws. This is including labeling. That's a big one. You'll hear that over and over. Um, Heck, even from even the way that a sample is passed in between collection and to us also counts as pre-analytical. Analytical is the moment that we begin the test on that sample, and post-analytical is once that reaction is done, then it's the reporting onward. Last but not least, medical history and physical examination are important in heme for a proper diagnosis. I cannot stress this enough. A lot of the stuff that you see here is supported by physical examination and medical history. Um, some things just based on physical examination alone and based on medical history could still be up in the air. So that's why we provide laboratory testing to help support the medical history and physical examination. Without the lab results, like I say, doctors are just guessing. So, doctors will order two forms of testing, or rather, two types of testing. First is a screening test. This is testing that is used to identify potential disorder in any asymptomatic patients. If your doctor has ever ordered you a complete blood count or a CBC, and you know that you haven't had any blood issues, or you didn't know you had any blood issues until they ordered it. That's the purpose of a CBC. It helps tell the doctor very general information about your current condition, and it could provide important clues for them to start ordering reflexive testings. So, based on the results of your screening test, your doctor can then add additional testing that is a bit more specific to help identify a bit more specific conditions if they're trying to rule things out. And these are tests that are usually only added on when your screening test turns out abnormal. Most doctors, they don't jump into this straight away. Um, You'll hear about electrophoresis, flow cytometry, bone marrow testing, marrow differentials, hemoglobin solubility, etc. Um, just understand that the screening test is what doctors are going to want to order for most of their patients to keep a good eye on them. Reflexive testing is when they notice something's weird with the screening test and they want to find out why. And that's the end of that presentation. All right, now we're going to move on to the next one, which is cellular homeostasis. Again, follow the objectives. If you can do this, you'll be fine in these lectures. So now we're going to talk about biology in general. if you're if you're like, oh my god, I've already been through like 4 years of this in school, why do I have to learn this all over again? I promise you, I'm not doing this for fun. It's important concepts for you to understand because all these lead into either oncology or cell development or immunology. You just need to know the basics, and I cannot stress this enough. If you can't understand the basics, I cannot expect you to understand the advanced material. So don't be afraid if you weren't so good in biology. This should help catch you up. So, for any organism to function, and humans included, there needs to be a correct amount of cells that are functional. If you have too much cells or too little cells, then that's a problem. Or you could even have the right number of cells, but if they don't work, that's a big problem too. So if any sort of this is out of whack, then that can be associated with certain disease states. So homeostasis is the carefully regulated process to help drive organ functionality, made up of these three steps. First, proliferation. Second, differentiation. And three is cell death. If you understand homeostasis, you can understand normal physiology. And when you understand normal physiology, you can understand cancer formation. And that's the big theme that I want you to understand. Okay, look, I'm sure a lot of you have already heard this. In case you haven't, we're just going to go over this real quick. Eukaryotic cells are cells that have a complex cellular organization that is made up of your nucleus, organelles, and a plasma membrane. This also is all suspended by a cytoplasm that is a liquid-ish that contains aqueous proteins, carbohydrates, fats, inorganic materials, and organelles. If there's sort of any sort of abnormality in a eukaryote cell's morphology, then that could help indicate a disease. Um, this will be talked a lot about in genomics if you're really interested in that. Um, but just sort of understand that if the cell ain't working, you ain't working, and that ain't great. A cell membrane's job is to be a physical barrier that helps differentiate your cells from the environment that surrounds them. The cell membrane function is to also help regulate ions, nutrients, and wastes. Proteins that are embedded in the cell membrane can help drive reactions by drawing these reactants closer to one another. And also because it's able to regulate ions, nutrients, and waste, it can also maintain pressure in ion gradients. That wall, that membrane that we talk about, is made up of a complex array of lipids called the phospholipid bilayer. Phospholipids are amphipathic. That means they are both polar and nonpolar. So polar, or this hydrophilic head, faces the cytosol or your environment because, again, that's like majority water. Polar likes to be with polar, so water is polar. Those two will like to hang out with each other. Then you have this nonpolar or hydrophobic tail that faces the inside of that bilayer. It doesn't like water, so it'll stay away from it. And these tails will face each other in this lipid bilayer. Um, so, but this isn't like a wall, right? I don't call membranes walls. If you have a cell wall, you're a plant, but you're not a plant, presumably. So, Your membrane is designed to move, not so much that it's gonna wobble so much that it falls apart, but it does move. It's a fluid almost. And the things that help regulate this fluidity are made up of four major phospholipids. I'm not gonna say their full name. Please do not memorize their full name. I'm just gonna go by their initials. But if you can kind of sort of see where the letters are coming from, that's good. First is PE and your PS, which is made up of your inner layer. PC and SM is made out of your outer layer. All these cells work together to help provide the right amount of fluidity, permeability, and the framework needed. So remember, like I said, it can move. This membrane can move around. Because of that, it always has this asymmetrical distribution between lipids and proteins. Um, when the cell membrane moves, it can respond to signals from the cell. And this is really important in apoptosis, where it sort of rearranges its walls to allow white cells to go ahead and start eating it up and mediate the cell's destruction. A major component of the cell membrane is also cholesterol, and this helps provide fluidity. Now, you just want this to be just right. If I got too much cholesterol, then the cell membrane will fall apart. If I got too little, then it won't move at all. And if you ever have snapped like a twig, then that will happen to the cell membrane if it runs into sort of any push or shove. And we don't want that. Um, like I've been alluding this entire time, cell membranes not only have lipids, but it also has proteins. These proteins could be for a lot of different things. They could be channels for ions, vitamins, minerals, or other substances. They could be embedded on the surface, or they could be integral to the entire plane of the plasma membrane. It could just be attached on the outside or on the inside of the plasma membrane. It could either be facing the outside or the extracellular or the inside, which is the cytosol side of the membrane. And proteins will help carry out the functionality. So it does a lot of this selective permeability, structural support, substance transport, enzymatic activity, and cell signaling. Whew! Proteins can do a lot. Um, they can also be modified with a carbohydrate. When it's modified with a carbohydrate, we call it a glycoprotein. Glycosylated proteins help play a role in cell signaling, recognition, and cellular adhesion. If you hear a lot about antigens, this is what an antigen is. It's a glycoprotein. Now, your body can use it as a way to help identify non self and self, but cancer does this little tricky thing where normally it's supposed to look like a thing to be destroyed, but can it actually manipulate its own glycoproteins and its own lipids to help sort of mask it itself as a normal cell to your immune system. And this is what it all looks like. I don't I don't really know how true um your body looks like your cell wall membrane looks like a blob, but this is sort of an example of your proteins and where they can fall within that membrane, and this is that phospholipid bilayer. Remember, the hydrophobic polar heads will face the outsides, and the hydrophobic tails will face each other. The cytosol or the cytoplasm is where most of your cellular and metabolic activities occur. Some protein synthesis will happen here, and it's where translation happens, growth, motility, and phagocytosis, etc. Cytoplasm is made up of an aqueous fluid that's made up of solutes, ions, proteins, and organelles. All these help that cell live. The cytoplasm composition will differ based on cell lineage and the degree of the cell maturation within that cell line. Again, important to know because we do look at the cytoplasm and what it contains inside of it to help determine normal versus abnormal cells. And you'll learn more about that when we go over cell development. This is a list of all the organelles in any eukaryote at a time. Um, I would say if you don't recognize the words, just give it a brief scan over. Um, but we're not going to go super deep into this. This isn't a molecular class. But just understand that these do exist. Um, all right. The big honcho, the brain of the cell, or known as the nucleus. The nucleus contains the genetic material, DNA, and is responsible for the regulation of your cell activities. When we say the genome, that is the total genetic information that's found inside of an organism, which is arranged in chromosomes. Um, if you some cells you might notice have this little fading this little like circle spot that I've highlighted in red here. That's called a nucleolus. It's an immature pocket that's found within the nucleus that's containing RNA. If you see this, then that means your cell's still developing and growing. And if this is very specifically to hematology, if you see a lot of cells with a lot of these nucleoli, that can be a blast. All right, so that was the basic cell parts. Now we're going to move into cell replication. And DNA organization. DNA, it's not like loose spaghetti that's just kind of floating around in your body. Um, it's wrapped around what we call histones, which then helps form a nucleosome. This nucleosome can then be further condensed into chromatin, which that can then be further coiled up into a chromosome. When we need it to be transcriptionally active, we're going to unwind a bit from this chromosome, from the chromatin, and just a bit from the nucleosome, so that way we can access this DNA that's sort of floating. Um, which is when it's transcriptionally active, DNA becomes referred to as euchromatin. When you look at a cell under the scope, you're going to see that the nucleus is really light and fluffy. It looks, you'll see the terms lace-like. Um, I like to think of it like those fuzzy carpets that you see in those furniture stores. And it's just going to be dispersed out the nucleus that way. Um, it's also unmethylated and highly acylated. This just basically means that it's not super modified in a way that your cell wouldn't be able to read the DNA anymore. We can also turn it inactive. So when we're no longer using it, we coil it all back up, back into this chromosome, and that becomes heterochromatin. Ooh, the central dogma. You probably heard a bunch about this. If not, that's okay. It's this idea that we can go from DNA to mRNA to protein. And this is how your body develops and grows, and how your cells develop and grow. The process from going from DNA to mRNA is called transcription, and mRNA to protein is called translation. Though this is a little bit outdated because I believe most textbooks will show this arrow going both ways. Maybe not back to mRNA, but between mRNA to protein, these can go back and forth. DNA and gene expression drive cellular morphological functions and differences. I cannot stress it enough. If your blueprints are messed up, then your mRNA is going to get messed up. If your mRNA is going to get messed up, your proteins are going to get messed up. Um, Genes are the specific locations um, in the DNA, and when we want to talk about that specific location, we call it a loci. mRNA are the genes that are organized into exons that are separated by introns. Exons are the parts where your cells actually read to translate into proteins. Introns are bits of fluff that tell it to stop or just not read. We can also change up this mRNA as we need. We can add in introns or intron splicing. We can add a 3-side poly A tail or the 5-methyl G cap. That's a little too advanced for hematology to just understand that we can change it up a bit once we need to. Then once we need to translate it, then this will pop out of your nucleus and into your cytoplasm, and then this um, Your translated RNA will interact with the ribosome complex. That ribosome complex will then match up everything together and form these amino acid polymerization and protein creation. You don't need to go super in depth into that. I just need you to understand sort of very generally the central dogma that we need to go from DNA to mRNA and protein. And if any one of these steps go wrong, then that affects the other. Tissue homeostasis, like I said, homeostasis is the maintaining of a healthy body. And we need to make sure that we have the right amount of cells that are functioning in order to help maintain organism function and viability. Every single body cell in your body, including blood cells, follow the same exact pattern. Proliferation, which helps replace lost or functionless cells, differentiation and maturation of these young cells to become specialized, to have a specialized function, which helps provide variety, specialization in any complex tissue or organ function. And when we are done with that function, it will eventually die and be eliminated, which is an active process. That way, we can free up more newer and better cells to help replace the ones that have been eliminated or died. When we get out of any sort of whack between these three steps, then you can enter in a disease state. This can happen from tissue atrophy, loss of function, excessive growth, or hell, cancer. So, the cell cycle, we are going to talk about sort of the two stages where it is your cell is just growing before it needs to go undergo mitosis. So we talk about these stages from G1 to S to G2 to M. Um, Interphase is where your cell is not actively dividing and it's not really sort of growing or changing. So this sort of part of this cell cycle, or all phases, are interphase. And each time your cell undergoes this sort of cycle, and especially at the end of mitosis, your cells lose pieces of DNA called telomeres, which serve as a checkpoint regulator. And if you run out of the telomeres, that tells your body to send that cell into G0 or quiescence. G1 is just the cell growth or synthesis of enzymes, protein, or organelles needed in order to undergo cellular function. And it does also need to check in and make sure that the cell is the right size. And if it is the right size, then it can advance into synthesis or S phase, where we replicate all our genetic material. We double check to make sure that DNA has not been messed up, because again, if we mess up the DNA, then we mess up the mRNA, which we then mess up the proteins, and that is very bad. So we want to make sure that no errors occur. Then last but not least is G2. It's just expanding one more, your cells are just expanding one more time to undergo mitosis. Mitosis is that other beautiful phase, and this is what we talk about. Prophase. Chromatin condenses into chromatins, your nuclear membrane breaks down, and microtubules start appearing. Metaphase. Your chromosomes are now starting to align along the poles, along the middle. Then anaphase, your microtubules grab onto those chromatids and they start pulling them towards the poles. Telophase, where now these chromosomes have now moved to the opposite ends of the cell and the nuclear envelope reforms. And last but not least is cytokinesis, where the cytoplasm starts pinching in the middle until it pinches enough that we separate out into two different cells. For anybody who is super visual learner, then this is a really nice, simple, easy diagram to follow along. So, like I said, when your cell's undergoing through its cell life, we don't want it to make mistakes, because when it makes mistakes, then the cell either becomes unfunctional or becomes rapidly prolific, which turns into cancer. And we really don't want that. So we have a few checkpoints. we have two types of cells or two types of genes whose job is to stop cancer from forming. So, first one is called proto-oncogenes. It's quite literally meaning before cancer genes. Proto-oncogenes are like the green signal to help cells advance in the next stage. When these genes are red and activated, then they stimulate mitosis and cell proliferation. Tumor suppressor genes tell the cell to stop developing, stop going. Both of these in balance should lead to a healthy tissue. But if we have any defects at either of these checkpoints, then what should happen is that the cell cycle either A gets to fix the problem, or two, we can't fix it, therefore we must undergo apoptosis. If we can't stop the cell cycle, then that's a hallmark of cancer. So, how do we get to cancer from what do you think would happen to these two genes in order for it to turn into cancer? Now is probably a good time to pause and think. What I'm asking is, um, what genes would need to be turned off and on in order for cancer to occur? All right, just gonna give a few seconds. All right, proto-oncogenes would have to be turned on permanently, and your tumor suppressor genes would have to be turned off. So what happens is when we have these stuck on, this go signal stuck on forever, and there's nothing to stop it, then think about what happens at a traffic light. If you just have a green light going on forever, then every single car is going to keep going and keep going and keep going and keep going and keep going with no stop signal, so your cell's just going to keep multiplying out of control. So, proto-oncogenes need to be turned on permanently, and tumor suppressor genes need to be turned off permanently in order for cancer to occur. If one is just turned on permanently, but the other one still works fine, then we're still in check. So both of them must need to go wrong in order for them to become oncogenes. So proto-oncogenes can then become mutated to become oncogenes. So like I said, oncogenes will need to be stuck on. It'll just be told to keep going and keep going and keep going. When a tumor suppressor gene it loses mutation mutates, it'll lead into a loss of function. So and this is um again we need both of these to happen at the same time in order to get to cancer. By the way, this one's a dominant mutation, which basically means only one allele needs to be lost in order for for proto-oncogenes to become oncogenes, and tumor suppressor genes must receive two alleles to be mutated in order for loss of function to occur. Alright. Differentiation is the process that generates cell diversity in each population throughout the body. Differentiation is driven by cytokine stimulation. So cytokines will bind to cell membrane receptors, which then start changes to gene expression and create cell lineages. This process must call for commitment before differentiation occurs. Basically, commitment just says that, let's say they have two daughter cells that come from the same cell lineage, but they must commit to two different routes of differentiation. Commitment and differentiation is both driven by cytokines. Just understand that once a cell has been committed, then you cannot jump over to a different cell lineage. So if I say that I have the stem cell that develops between a red cell and a white cell, that red cell, once committed, can't just jump over and become a white cell. Doesn't happen. Again, differentiation is driven by gene variations, which will then create different proteins. And remember that protein drives function. Protein changes will cause function changes. And a lot of the times we want these changes, but sometimes the changes can be really bad, and that's what happens in disease. Um, this is a wonderful little thing that helps drive diversity in our population. Epigenetics are stable genetic changes that are passed down through generation. So I talked about this gene silencing a few slides ago. Um, so that's what this is. These modifications that are carried down through um, the cell lineage, and that's done via methylation, formation of heterochromatin. So for hematology, commitment and differentiation plays into this role. All of these cells start from this multi-potential hematopoietic stem cell, a name that's pretty straightforward considering science terms. That stem cell then separates out between these two lines. So once this has become a common myeloid progenitor, it can't just jump over and become a common lymphoid progenitor. This megakaryoblast just can't become a myeloblast. So, because of the combination of commitment and differentiation, we get all these beautifully diverse cells that all work together and help maintain function in your body. And it's cool. It's really neat. Um, you'll understand this chart more as we go through class, but just understand that everything starts from one cell, then that one cell is told to become one of two things, and that continues on until the cell becomes fully mature. All right, and this should be the last concept before we wrap up today's slides. So we're going to talk about apoptosis versus necrosis. Apoptosis is, or like, when a cell eventually enters G0 or senescence, it'll exit the cell cycle and it'll perform its functionality until it dies. That means the cell will no longer respond to any growth cytokines or mitogens. And remember, this is not considered a part of the cell cycle. The whole point is that when it enters G0, it has exited the cell cycle. It is done developing and repeating that cycle over and over. So as cells age, they will lose functionality and eventually die by either apoptosis or necrosis. Necrosis is called cell murder. We really did not intend for it to die this way. Usually it's a very messy event that's induced by lethal chemicals, biological, or physical events. Apoptosis is cell suicide, where basically the cell tells the body that it's ready for its own destruction. Apoptosis is vital, at least at a regular rate. We don't want it to become too much because that kind of defeats the whole purpose of cell development. And we don't want to happen it for too little because, again, cancer can occur. So it's a structured and organized cell death. We want this to be organized. Um, Apoptosis will occur after three things. So, first is that other cells will start putting out these survival signal or lost survival signals to basically tell it to die. Cytokines like TNF or Fas ligands are these like death signals, so those will start increasing as well. And last but not least is the cell has experienced irreparable damage or stress. So because it is structured, it is organized, we need energy or ATP to initiate apoptosis. It doesn't happen spontaneously. It's a very well, beautiful, controlled process. And because it is organized, we don't cause damage to surrounding cells. A lot of the cells that your body contains becomes pretty damaging when it's no longer inside a cell. So we don't want that to just fly all over and affect other cells. We want this to be a very beautiful and slow and contained process. On the other hand, necrosis doesn't care. Necrosis will happen due to disease or trauma to the cell. Think like eating Tide Pods or something. I don't know what the bad thing to do nowadays is. Or you get a cold and an infection causes your cells to be irreparably damaged. Um, Something along those lines. It's unstructured, it's unorganized, or it's murder. And this will be caused by external stimuli like toxins or hypoxia or ATP depletion. It happens spontaneously. It does not require ATP. It's a very sudden and brutal thing to happen, and it's therefore an inactive process. So when your cells die via necrosis, nobody expects it, right? So there's nothing to clean it up. There's no white cell anticipating a cell cleanup. So your cells just kind of pop like a bubble. And like I mentioned, there's things on the inside of your cell that are helpful when inside of a cell, but no longer helpful when it's not contained. And that'll cause damage to the neighboring cells, which then leads to inflammation. And inflammation is a sign of disease, and that can be very bad. So, necrosis is what happens on the left. There's some sort of, so if we have a reversible injury and it can handle the stress, it'll come back to being a normal cell. But let's say that this injury becomes too much for the cell to happen. Well, then it starts swelling up and quite literally just popping like a balloon. It just goes everywhere. And there's a lot of contents in here that we don't want outside of the body, and your immune system is going to treat it like inflammation. um but in apoptosis, it's a very controlled process, but we do need energy in order to be able to condense it and break it apart and tell the right signals to the right cells that, like, I'm slowly fading away. So these apoptotic bodies will pop out as the cell dies, and these apoptotic bodies are cleaned up by your white cells. So, again, necrosis, uncontrolled, just wild. Everything goes all over the place. It's spontaneous, and we don't have energy involved in this. But this is a very controlled, heavily invested process that does not cause damage to the outside of other cells. Um, if you are the type of person who hates both voices and visuals, then this is a really nice Venn diagram between the two. Um, I make necrosis sound really scary just because it's an uncontrollable process and it just kind of happens spontaneously. But apoptosis, when increased or decreased, can still lead into disease states. Again, we want apoptosis, but we need it to be at a controllable level. If we have an increased amount of apoptosis, then it leads into disease. If we have a decreased amount, then we lead into other disease as well. It's all a balance between proliferation, differentiation, and cell death or apoptosis. And if we don't have any of those three in balance, then we as an organism can't live. So thank your body sometime, and thank biology for helping us understand this sort of basic concept to living. And that is the end of the slides. I hope you enjoyed this presentation.